In this module, we'll discuss the brain basis of behavior. We'll start with talking about the structure and the functions of the nervous system, and then we will talk about how this brain and the nervous system is organized and how this organization leads to complex behaviors. And then finally, we will talk a little bit about some really interesting aspects of the organization of the brain that lead to interesting conditions. This is because to understand what makes us who we are, we need to understand the physiological processes and the biological underpinnings of our thoughts, feelings, and behaviors. And every cell in the brain, every molecule in that cell in the brain contributes to how this system works, how the networks are laid out, and how the function of those networks ultimately result in specific behaviors. So what we'll be discussing right now will be the basis of all complex behaviors, of perception, of decision making, of intelligence that we will subsequently be talking about. The specialized cells that control our behaviors belong to the central and the peripheral nervous system, largely brain, the neurons, the nerves, and they can be studied at different levels. We can study the anatomy and look at the structural aspects of what the brain is. We can study the physiology and understand the functional underpinnings. Studying the embryology and the development of these systems, these networks, is the basis of how differences in individuals come about. Studying the pharmacology, the neurochemistry, and the drug action, critically important for our abilities to modulate these functions. And ultimately, psychology, which is the regulation of behavior that relies on all of these other things coming together. So while psychologists are mainly interested in studying the regulation of neural activity, understanding the nervous system per se, especially the brain at all levels of analysis, has led to many important insights. So as you probably already know, the nervous system's main component are the neurons, the cells where all the action is. These are the building blocks of all behavior. We have about 1 billion to 1 trillion cells that receive, integrate, and transmit information in the nervous system. They form what we call the neural networks that regulate behavior. An even larger proportion of cells in the brain are called the glia. They form the myelin sheaths, the insulation of the brain. They provide nutrients and oxygen. They support the neural structures and remove dead cells and pathogens from the brain. The glial cells are of many different types, and the functions of all of them together contribute to the efficiency with which the neurons work. As I said before, we have about 3 times as many glia as neurons, and scientists think that so far, glial cells have been thoroughly underappreciated. Every day, we find new significant contributions of glial function to our behaviors, and I'll talk about some of those at a later point. And finally, the brain function also relies on the rich vascular supply, the blood vessels that bring in the nutrients and the oxygen and take away some of the toxins and the metabolites. About 40% of the blood pumped by the heart goes to the head. The neural tissue is one of the most active tissues in the body. So any compromises in the vascular or the cardiovascular system can lead to compromised brain function as well. So as you can tell then, the structure of the nervous system is very, very critically important to the function of all of these different types of cells. Researchers have learned a great deal about the biological basis of brain activity due to the development of technology and advances in our ability to study the biological processes. For example, scientists have been able to study why particular drugs affect thoughts and emotions in specific ways. Let's view this video that shows some of these techniques, and then we'll talk more about the specifics of neural function.
The brain of a mammal is one of the most complex things in the universe, but studying brains has become easier thanks to some complicated high-tech equipment. This is the Tanagawa lab at MIT's Picower Institute for Learning and Memory, and this impressive machine is their 2-photon microscope. It has 2 lasers that intersect in the sample of brain tissue, They excite photons of light, which, when amplified, reveal intricate structures. The researchers take snapshots at a range of depths, then combine them to create a layered, three-dimensional image that reveals features of the spines that protrude from brain cells.
So a neuron is like a tree, and it has branches, and the branches get finer as you go farther out along the tree. And on the tree, you can imagine they're almost like little thorns. There's spines, and the spines are basically the sites of connections between two neurons.
The images allow researchers to determine the number and thickness of the spines. Studying them has revealed important structural differences between normal brains and brains with disorders like Fragile X syndrome. Then, in the electrophysiology lab, researchers subject tiny samples of brain tissue to small pulses of electricity to measure their conductivity.
And that's where the neurons communicate with each other. So if you have a defect in the spines, then you have potentially problems in communication between neurons.
Too much conductivity means an overload of stimulation, a problem researchers see in people with Fragile X and in some cases of autism. The busy connections make learning and attention difficult, and new, highly stimulating experiences simply overwhelming. That's what these big machines tell us. But for more video on how the Tanagawa lab is working to quiet these noisy brain signals, check out ScienceCentral.com's story, Reversing Fragile X.
So behavior then is controlled by signals in the neuronal circuits. The simplest kind of circuit is known as a reflex circuit. It consists of an incoming sensory signal carried by what are called the sensory neurons, or the afferent neurons, with an A, that is. They direct information from the outside world and pass that information along to the brain. For example, somatosensory nerves from your skin and muscles bring information into the spinal cord and the brain. This information then is relayed to neurons within the brain. They are called interneurons, neurons that communicate locally and process and integrate this information. Those signals are then passed on to the neurons or nerves that carry information to, say, your muscles. These are called the motor neurons. They can direct the muscles to contract or relax. So in the simple reflex circuit, for example, a pain withdrawal reflex, any pain signal at the level of the skin will result in signals through these three types of cells to pull the arm away or to pull the finger away, as shown in this figure. Let's look at a typical neuron. This is a schematic of a real neuron, and you can see that there's a rich branching at both ends. At the receiving ends, these branches are called dendrites and are busy picking up signals from as many as a few thousand neurons. These signals are then communicated to the cell body known as the soma, and there, if new electrical activity is generated, that gets transmitted down the long cable-like length of the axon, which conducts the signals away from the cell body into the terminal. This terminal is also branched, and at this point the cell may make contact with many thousands of other neurons. So neurons operate through electrical impulses and communicate with other neurons through chemical signals called neurotransmitters. In a more schematic diagram, you can see that the sending neuron has these dendritic collectors of signals. Then the cable, the axon, which is the conductor of these signals, and finally the terminal, the branch that makes contact with the receiving neuron. Notice that the cable, the axon, is actually insulated by these layers so that there's no leakage of the electrical current that's being passed up. As such, each part of the neuron is specialized for its function. At the terminal, the neurons form these bulbous bouton-like structures that are nodules which contain many sacs of neurotransmitters called synaptic vesicles. These vesicles release chemical signals into the synapse, the connection with the other neuron, when a signal arrives down the axon of that neuron. Electrochemical communication, then, is the hallmark of neural activity. Neurons communicate with each other using chemical signals called neurotransmitters and send signals from one end to the other within a neuron using electrical gradients. This electrical signal is called an action potential. The membranes of the nerve cells have special protein gates and pumps that selectively pump charged molecules, ions, in and out of the cell. This is usually referred to as selective permeability. These pumps place different types of ions, specifically sodium and potassium ions, either out or inside the cell, and that results in a net negative membrane potential inside the neuron. The potential is about minus 70 millivolts. So it's a really small charge difference, but it makes all the difference. Therefore, at rest, we refer to the neuron as polarized, meaning it is away from zero polarity. This polarization creates the electrical energy that is necessary to power the firing of a neuron. When neurons receive chemical signals from nearby neurons, excitatory signals depolarize the cell membrane, by which I mean that they reduce the polarization. This increases the likelihood that the neuron will go into what is called the firing mode. As the neuron gets closer and closer to the threshold potential, which is about minus 50 millivolts, the sodium channels open and let in a rush of positive ions. Subsequently, voltage-sensitive potassium channels also open, and this results in the firing of an action potential. The action potential is a stereotypical set of electrical changes like the ones shown in this graph here. The membrane potential typically very rapidly depolarizes, and then as the ion channels begin to close, first the sodium, then the potassium, the cell repolarizes and the potential drops to the negative 70 and sometimes a little beyond, which is referred to as the hyperpolarization. As a result, what happens is a rapid reversal of potential. This really brief, about millisecond and a bit long process is what we call the nerve impulse. When a neuron fires, the inside of the neuron becomes slightly more positively charged than the outside. After firing, the inside of the neuron returns to its slightly negative resting state. Typically, a neuron will either fire or not. This is known as the all or none principle. Once the cell goes past threshold potential, an action potential is generated, and the action potential always has the same profile for that cell. The depolarization within a neuron diffuses through the cell structure and the cell body, but is also propagated actively by the presence of the ion channels and the pumps at various locations on the axon. These pumps are concentrated at what are called the nodes of Ranvier, which are the gaps between the insulation, between the myelin sheets. Myelin prevents the movement of ions away from the membrane in the extracellular space. It also reduces the capacitance of this nerve membrane. So the fact that these ion channels are all concentrated at these nodes makes the action potential travel much faster in myelinated neurons than in non-myelinated neurons because it's essentially jumping from node to node. This is referred to as saltatory conduction. Action potentials are typically generated in the cell body and always move away from the cell body towards the terminal boutons. Insulation provided by the myelin sheath enables the action potential to skip quickly along the axon. This saltatory conduction is a hallmark of good conduction. Losing the myelin sheaths, often referred to as demyelination, can result in slowing down of neural impulses, like in the condition multiple sclerosis, which results from a demyelination of axons and compromises motor coordination. Once the action potential reaches the terminal bouton, it causes these neurons to release neurotransmitters. This happens at a specialized structure called the synapse, the seat of all neural communication. At this place, the presynaptic cell is specialized to release neurotransmitters, while the postsynaptic cell, the cell that is picking up the signals, is specialized to receive these chemicals. The postsynaptic cells have specialized proteins called receptors that bind to the neurotransmitter, and this binding results in either excitatory or inhibitory signals within that cell. This once again is due to the opening and closing of specific ion channels. Let's take a look at the next video, which shows synaptic transmission in detail.